thanks for all of you coming today. Thanks for ARM for the invitation and opportunity to present here. It's like everybody, nice to see mostly smiling faces or masks in a real audience these days. So I'm going to talk to you today about ShorelineBiosciences. This is a company that I co-founded, and I'm currently serving as Chief Scientific Officer. This is an iPSC-based company looking at allogeneic cell therapies, really focused on two products, two cell types, natural killer cells and macrophages. So this is our founding team. We founded June of last year. Like a lot of folks here, this really has developed over the COVID times. Cleanthus Anthopoulos, who's a serial entrepreneur, is our chairman and CEO. I'm serving as CSO. Usually I have to disclose my industry ties. Now I have to disclose my academic ties. I am still a professor at UC San Diego. And then Bill Sanborn is our chief medical officer, was also at UCSD, retained some ties there, but really has a strong history in inflammatory bowel disease. And we've really filled out an excellent leadership team over this past year. So again, really, we use the iPS cells as a platform for our core technologies. And as shown on either side here, again, we're really focused on both iPS-derived natural killer cells as well as iPS-derived macrophages, the NK cells really being our lead products. And we can use the iPS cells, again, as a platform to then add in different technologies, which I'll explain, which include optimized chimeric antigen receptors, other versions of CD16 to mediate antibody-dependent cell-mediated cytotoxicity, and a system of universal engagers that add to the flexibility and multi-targeting of these cells. We also have the ability to use the iPS cells to engineer macrophages with CARs and other edits. So really, this is the platform that was developed in my lab where we're able to focus on iPS cells. We can make our gene edits, as I'll explain. The main edit is the deletion of a gene called CISH, which I'll explain in a minute, which leads to markedly improved NK cell function. So we can make that knockout and or add in our CARs or other engagers of interest, derive a clonal cell population that we can look for off-target effects and so on. This then undergoes the NK cell differentiation, expansion, and cryopreservation. So as I'll explain, with the CIS knockout, we can develop NK cells, which we think will be a best-in-class product, as we've shown that this leads to improved persistence and an improved metabolic profile, reduced exhaustion, and a so-called polyfunctional cytokine profile, which I'll show you more about. So this just shows a little bit of the biology. The gene CISH encodes for a protein called CIS without the H. This is a negative regulator of IL-15 signaling. So as has already been noted, a lot of companies are looking to improve NK cell activity by expression of IL-15 in their cells. Here, we essentially remove the negative regulator of IL-15. So normally, IL-15 signals through JAK-STAT signaling that leads to upregulation of CISH expression, which leads to ubiquitination of these JAK components, JAK-STAT components, which normally would turn off IL-15. So we have shown that by deletion of CISH, you essentially take your foot off the brake of these cells. This leads to increased proliferation and activation. Again, an improved metabolic profile with both enhancement of glycolytic activity as well as oxidative phosphorylation. This is an mTOR-mediated process that leads to improved anti-tumor activity. So again, we use this CISH deleted iPS-derived NK cell really now as the engine that's going to drive our NK cell product that we can build in other edits. And again, as we've shown in this paper published last year from my group, these now have increased sensitivity to IL-15, the improved in vivo persistence, decreased exhaustion, improved cytokine release, and we think that this leads to improvements overall in the immunosuppressive microenvironment. This is one example of this technology. These are using a AML cell line in an immunodeficient xenograft mouse model. So untreated, the tumors grow. If we use the typical unedited iPS-derived NK cell, you do get some modest anti-tumor activity. This is with just one injection of the NK cells. But now with this cis knockout iPS-derived NK cells, we can completely clear the tumor in 60% of the mice, go out 100 or more days, and there's no signs of disease relapse. And again, we can show in the paper that this is due to the increased levels of NK cells, as well as increased homing of the NK cells to the tumors. So now, again, we use this as a platform to add specificity through CARs or ADCC. We can improve persistence further by addition of different IL-15 molecules or decreasing the immune recognition of these cells, other methods to enhance the activity of the iPS-derived product. So again, one of these is development of NK cell-specific CARs. This is something that has done previously in my lab, where we found that NK-specific CARs work much better, especially in vivo, compared to T-cell CARs expressed in NK cells. So we're now looking for ways to further optimize this by screening other co-stimulatory molecules within a NK cell platform. We also are testing macrophage-specific CARs, which similarly we think will further optimize this strategy. The other way that we can enhance this is not only build in a NK-specific CAR, but combine this with so-called universal engagers. This is in collaboration with Travis Young's group at Scripps Caliber, who developed a so-called switchable CAR technology that's now in clinical trials as a autologous CAR T cell. The strategy is you develop a CAR that is specific for a certain peptide, and you can then engage, implant that peptide in these soluble FABs or other targeting constructs or binders with different tumor-associated antigens. So in this way, you can add either multi-antigen targeting, or you can actually switch your targets over time, all using the same engineered NK cell platform. Another strategy, as I mentioned, is we're also developing iPS-derived macrophages. These are becoming much more interesting, both for cancer therapy and non-malignant disease. So if you're not familiar with macrophages, they're divided into a M2, which is a pro-inflammatory population that typically produces inflammatory cytokines. There are also M2 macrophages, which tend to be anti-inflammatory or can mediate tissue repair and regeneration, and these produce cytokines that are typically anti-inflammatory. So we've developed, using our iPS platform, we can make macrophages and skew them either towards the M1 or M2 phenotype. We're now, as mentioned in the process, we can express CARs within these Ips Drive macrophages and or combine these with antibodies. The cells normally express FC receptors, or we can, again, engineer in other molecules to bind to antibodies or other binders. So again, really now using the platform that Shoreline has developed, using Ips cells, which form the basis of doing these genetic modifications. This is technology my lab has been doing successfully for really the past 20 years of work with human pluripotent stem cells. We've developed efficient strategies, and this has been even further optimized within Shoreline for NK cell differentiation after the cis knockout, and so we can really get a large-scale production of homogeneous, 100% gene-edited Ips-derived NK cells or macrophages. And as I think been, was discussed this morning and in other sessions, this scalable process really begins to lower the cost of this type of cell therapy, though it is, the manufacturing and CMC is still critical for this process to be successful. Since the founding of Shoreline just over a year ago, we've engaged in two key strategic partnerships, one with Beijing, which is funding additional research and development and has, as I'll show you, markedly expanded our pipeline into additional blood cell cancers and solid tumors. And the rights for these are divided between the U.S. and Canada can develop, Shoreline can develop, and Beijing has rights for Asia. We also have a partnership with Kite, also for R&D and for development of 3 to 5 additional programs primarily focused on hematologic malignancies. So this is really our pipeline for Shoreline. As was mentioned, we were really developed, focused initially on the cis knockout NK cells, and our initial product will use this to target AML. This would actually be without additional CARs or engineering beyond the cis deletion. There is a strong history of using allogeneic NK cells, has been discussed, that are highly effective for relapse refractory AML. And we think by having the cis engineering, where you have endogenous increased activity due to IL-15 signaling, that this provides a potent anti-AML strategy. And we hope to have the IND for that by the end of next year. We're also, again, based on different CARs as well as addition of antibodies able to target different hematologic and solid malignancies, as well as T-cell lymphomas. Following this a little bit further back in our pipeline will be the development of the Ips-derived CAR NK cells. Then again, with Kite, they are looking at using our cis knockout technology combined with 3 different CARs for hematologic malignancies. And then with Beijing, again, is using the cis knockout, looking to target either solid tumors or hematologic malignancies. So that's really where we are with Shoreline. We have, I think, a very ambitious program, but I think we have all the know-how and a great team now in place to get this done, and with the goal of IND by the end of next year. I guess I do have exactly 1 minute for questions, if anybody has them. So, yes? The goal, and we have some data and strategies, is that we can essentially lock them into one phenotype or the other. And I didn't mention, since you mentioned the macrophage, again, we can add CARs to this. And I think, while there is interest in CAR macrophage for anti-cancer therapy in and of themselves, these obviously are very infiltrative into tumors and can be combined with NK cells or other agents. I'm not, sure that in the end the CAR macrophage in itself will be the sole therapy, but it is a great tool to, again, turn cold tumors hot in this setting. And I guess that's it. Okay, thank you very much.